Hello, my name is Lotte Van Peteghem and today I would like to talk to you about the synergy between carbon capture and utilization and microbial protein production. Let's go for a carbon neutral food chain. First of all, I regret to announce that we're not eating these delicious Turkish typical foods together. But a good thing is that we're not emitting 15 kilograms of CO2 while eating this köfte. Or we would be emitting a bit less, but still 1 kilogram of CO2 when enjoying this delicious fish sandwich. In essence, this CO2 actually first is incorporated into plants. So it comes from the air, incorporated into plants, and then the lamb is eating the biomass. But while processing and during the growth of the lamb, a lot of this CO2 gets lost. And this cycle actually is very similar for the chicken and for the fish that ends up on our plate. If we look at the full picture, 25% of the anthropogenic greenhouse gases is emitted thanks to the food production industry. 50% of our habitable land we use for agriculture, and 70% of our freshwater consumption can be attributed to food production. And when we look at the future, our world population is expected to grow, and it's also expected that more and more people will switch towards protein-rich diets. So this protein demand is actually going to double by 2050, at least according to the predictions. Now, is there any alternative? The alternative that I would like to propose you is microbial protein. What is microbial protein? Microbial protein is in essence biomass that is produced by bacteria, filamentous fungi, microalgae or yeasts, and it's rich in protein and can be consumed by animals or humans. When we look at, for instance, the land use of these microbial protein, here an example where they're produced by using photovoltaic energy, and we can produce 15 tons of protein per year on 1 hectare with this process, while for soybean we could only produce 1 ton of protein per year. So it is very efficient in land use. When we look at the water footprint of microbial protein, I first have to tell you, feed, kind and quorn are actually two commercial microbial protein products, where quorn is the one that can be used for human consumption, and feed kind is one that is used in fish meals, for pets, for chickens, for porks, any kind. And when we compare this water footprint with, for instance, soybean, it's quite similar and it can even be better. And it's much better than pork or beef. Now, when we look at the carbon footprint of feed, kind and quorn, it is similar to the one of pork, but it still cannot compete with soybean, because soybean is a plant, it incorporates CO2 in the biomass, while these are heterotrophic processes, aerobic heterotrophic processes emitting CO2. And it's actually this that inspired us to present you a bit of a different system. So what do we want to do? We want to use carbon capture and utilization. So we want to start from CO2 point sources, CO2 and off gases that is wasted, combine it with hydrogen and convert it into commodity chemicals. As such, carbon capture and utilization. Not a new processes, more and more projects are being launched like this. However, there's two big challenges related to this technology. The first one is a low market potential of these commodity chemicals, and the second one is the economic competitiveness. So, for instance, when you want to produce methanol from CO2, you will need 3 times as much money in comparison to when we would do it from natural gas, and the same goes for formic acid. Now, CCU, it can be combined with microbial protein production. We use these commodity chemicals as an organic substrate for microbes. Microbes incorporate them in their biomass, and the biomass is used as feed or food. Now, it's an aerobic heterotrophic process, so a part of the carbon is also respirated, so emitted via the off gas as CO2. And the idea is then to send the CO2 back to the beginning so that we have a very efficient use of our carbon. Great system, if you would ask me. However, there's still a few questions that should be answered. A very first one is actually what kind of substrate is now the most interesting one? And more specifically, what metabolic pathway is the most interesting in terms for the production of microbial protein? How much energy do we need? Because we can say that we use wind and solar, but in the end, the electrical energy that we produce today is not all wind and solar. So there is a carbon footprint of this electrical energy. Can we reach carbon neutrality, and what are the bottlenecks of this technology? Our electric requirement to produce biomass is calculated based on the energy that was needed to produce a substrate, the energy that is needed to produce a biomass, and the electrical energy that was needed to convert the CO2 coming out of our microbial protein reactor back into substrate. And this CO2 off gas is actually very rich in CO2. It contains very little oxygen, about 2.5 to 5%. And why is that? Because we're using water electrolysis to generate the hydrogen that is required to produce a substrate. And simultaneously, oxygen is also produced. And the amount of oxygen that is produced here is sufficient for the growth of our microbes in the microbial protein reactor. So the oxygen production is enough for the aeration, and we can use pure oxygen to aerate our microbial protein reactor. Looking at the numbers, it turns out that acetic acid and ethanol are the most interesting substrates from an energy requirement perspective. Also, the RUMP cycle, when we use methanol as a substrate, isn't one to consider. There is a large deviation depending on the metabolic cycle. So it is crucial to also look at the kind of metabolic cycle that your microorganisms are using to see if it's actually a good substrate or a good process or not. When we look at the technology readiness level of carbon capture and utilization technology, for formic acid and acetic acid, the technology readiness level is 3 to 5, and for methanol and ethanol, it's 6 to 7. We haven't reached 9 yet. So I expect that this energy requirement to produce your substrate could actually still be reduced when we reach more matureness of this technology. And this energy requirement for your substrate production is in most cases also the largest contributor to your total electric requirement. Now, for the carbon footprint of our microbial protein, we'll look at the CO2 that was emitted via electricity, and herefore we used the average CO2 emitted in Europe per kilowatt hour. So here about 0.3 kilogram. The CO2 in the effluent, and this is unused substrate, lysed cells, dissolved CO2. And then finally, there is actually also a negative CO2 value because we actually do capture CO2 via the CCU process, and that's this darker color. We did also make a distinction between this electricity footprint in 2019 and the one that is expected in 2030, because the European Union is invested in decarbonizing their electricity sector. So we actually expect to have only 0.1 kilograms of CO2 equivalents emitted per kilowatt hour by 2030. And this actually means that acetic acid and ethanol, or definitely here acetic acid, could be carbon negative in 2030. Also, ethanol could be very close. And there's another side note that I should make here, is that when we look at the protein content of beef or pork, this is 16 or 10% of your total weight is protein, while for microbial protein, this varies between 40 and 70%. So then the impact is even bigger. Also, when we look at the CO2 emitted for 1 kilogram of pork or 1 kilogram of pork or 1 kilogram of beef, there are some scenarios, definitely in 2030, that go below also the pork. Water electrolysis requires a lot of water. However, when we look at the stoichiometry of, for instance, the substrate production of ethanol, there is water produced. The same goes for methanol and acetic acid. Also, when we look at the stoichiometric reactions of the biomass production from these organic substrates, there's water produced. And purely basing ourselves on this stoichiometry, we calculated that, for instance, for methanol, more water is produced than the one that is required for your water electrolysis. For formic acid, this is very equal. Acetic acid, there's still a slight gap. Now, this water that you then need for your water electrolysis could actually be recovered in the downstream processing of your microbial protein production. So for instance, here the wastewater after centrifugation, or the water that comes from your drying process. If we manage to get this to a good quality, we could actually substantially reduce the water footprint of the overall microbial protein production. I said in the beginning that the commodity chemicals produced via CCU have a low market potential. Well, actually, when we're aiming at feed and just feed, I'm not talking about food, we can increase this global market by up to 4 orders of magnitude. Now, looking at the market value, so the cost competitiveness of our CCU product, also here feed can increase our revenues given that there is a higher market value of our feed in comparison to these other now commodity chemicals produced via CCU. What food, for thought, would I like you to take home, not the Turkish food, from our presentation? What I want you to take away is that the oxygen concentration here of gas is, although very low, can still be an issue for your CCU. Will we have to remove the oxygen before? Can we find CCU processes that can deal with the oxygen? This is something to be investigated. Water recovery from our downstream processing will greatly help us to reduce the water footprint of our microbial protein production. And then in 2030, we could actually reach a carbon negative protein production process for instance, acetic acid. The market potential of CCU coupled to microbial protein production could be increased up to 4 orders of magnitude, and the product value can be increased almost 7 times. Now, what is left is thanking my lovely colleagues and co-authors of this study. You can reach me on the email here and or via the websites on this PowerPoint, and then I would also like to thank the institutions and company ArcelorMittal that made this study possible. And thank you, of course, for listening.